Yes, a warm welcome from me as well. My name is Roland, as Janusz just introduced me, and I'm very glad to walk you through what ChemAxon has been doing in the field of biomolecules recently. I hope that all of you will understand me well. I think Janusz is making sure that everything is going in the right pace, and he'll kick my leg if I talk too quickly or something's not right on the audio. So, for those of you who have been with ChemAxon for a while, you'll probably know that our entire product portfolio, to a large extent, is based on our JChem platform. For those of you who are not familiar with that, I'd like to spend a few words on what JChem actually is. As you can see on the screen, we consider JChem to be largely at the core, at the heart of our product portfolio. JChem itself offers you the leading chemical database search engine, also as an Oracle cartridge, and is largely a suite of integrated tools around handling chemical structures and chemical structure data. JChem is very customizable, very feature-rich. It's efficient and scalable, even up to the cloud environments, and is very accessible depending on your experience, ranging from kind of integration into end-user application for scientists, as well as toolkits providing you the opportunity to create chemical intelligent applications via our APIs. I will not go through all of the features, but giving you kind of an idea about where JChem is applicable, we offer the import and export of a large variety of chemical file formats, mol files, SMILES, our proprietary Morbid format. We also support kind of more high-level UPEC names, traditional names, and INCI identifiers for our chemical representation layer. We offer a connection to practically all major database engines. There are Oracle, MySQL, Postgre, IBM DB2, Derby, and others. For some of them, we even offer direct cartridge support. Oracle, for example, I think we are working on a PostgreSQL cartridge as well. And as a platform, JChem is largely operating system independent since it's a Java implementation. We offer for the Microsoft Windows environment a .NET API, and we also are offering JChem in the form of web services. It was SOAP web services in the past, but I think we've largely finished the switch to the RESTful API by the time we are talking. So that's all neat, and that's all fine, and that offers us a great way to deal with chemical structures on kind of the atomic representation. But we have been challenged in the past quite often with biotherapeutic entities which cannot be handled that easily by the classical cheminformatic tools at all. What you see here on the screen are, in fact, different representations of the very same molecule, Erythropoietin. Some of you may know this as EPO, very prominent in the world of cycling sports and other activities. You see a three-dimensional structure representation of the protein core together with the glycosylations on the protein core on the left-hand side. Then you can see a schematic representation of the very same molecule indicating how the glycosylation patterns are actually built from individual saccharides units. Then you could also look at an all-atom representation of one of the glycosylations on the right-hand side. And last but not least, as it is a protein-based molecule, you'll find the sequence of the individual amino acids on the bottom part. And you can already see there's a lot of kind of different information in the individual layers encoded of this protein. Within the sequence itself, you can also see that I've highlighted a few parts of interest here in color. So the green parts are usually amino acids which are truncated away when the protein is actually maturing. Then the red coded amino acids are the ones to which the glycosylations are attached to. And I think the brown ones are kind of mediating or have a strong structural role in identifying which glycosylation is actually added to this particular amino acid. So when we are thinking about handling these molecules in a classical cheminformatics engine, then you'll see that you lose the context of all of the sequence information of the additional annotations you might want to keep if you work with these molecules. However, if you work in biopharmaceutical R&D, then to a large extent what you actually try to achieve is you try to find a pharmaceutically active compound, and you don't really care whether this is a small molecule entity or it's a large molecule entity. What counts is that it's actively modulating your biological target in a way that it's, for example, helping you to alter the disease pattern of a disease phenotype. So in order to achieve this, we've decided to take on an entire new product line which we think will extend our capabilities of the JChem platform to the large biomolecule level. So for this, let me briefly describe what this kind of biomolecule toolkit, as it is its current state at the moment, does for you. The biomolecule toolkit is a template-based kind of toolkit which allows you to achieve an all-atom representation of even large molecules, and at the same time it keeps the biological sequence information intact so that bioinformatics tools can operate on the molecules handled and stored by this toolkit. It provides a standardized representation of large biomolecules, and this is especially important when you look at the kind of boundary areas between the chemical and the biological world. So typically, of course, a protein is often not just a protein sequence alone, but as you've seen in the example of the Erythropoietin, there are glycosylations happening. There are other chemical modifications which may happen on the protein backbone, post-translational modifications, an example. All this kind of additional chemical information can be captured and respected by our biomolecule toolkit. The biomolecule toolkit is fully compatible with the OpenHELM standard for representing these large molecules. For those of you who have not heard about OpenHELM, OpenHELM is an initiative by the Pistoia Alliance, a consortium of cross-disciplinary organizations in the pharmaceutical space, and it aims at providing a kind of representation standard for large molecules. It originally came out of Pfizer labs, and it's running now as an independent project in the Pistoia Alliance. We do have a project homepage which is openhelm.org for those of you seeking out more information, but I can show that later on in the browser as well. And last but not least, we evolved the biomolecule toolkit now to a state that we can offer a preview version for exploration. So if biomolecule handling is of your concern, feel free to approach us, and I'm sure we can somehow set you up with an evaluation version of that toolkit and talk about further implementations. Feature-wise, it offers you storage of large molecules in a structured fashion so that it's actually in the database optimized for specific sequence queries, chemical structures queries, and other additional data queries you might want to run against a set of large molecules. As we design this as a registration toolkit, we offer you a way for canonicalization, meaning that you'll have a unique representation of a large molecule for a unique macromolecule topology. We calculate certain simple properties, for example, a molecular weight or an isoelectric point for proteins, for example. We offer you SOAP and RESTful Web Service API, and we do have something which we term a configural duplication check, so you can somehow increase the sensitivity of the duplication check. If you imagine that you have a protein which forms internal disulfide bridges, for example, then the exact duplication check will actually capture molecules which have the same sequence and same disulfide pattern. If you loosen up this check a little bit, we may actually find a duplicate just based on the sequence alone, not considering any further disulfide bridges in the molecule. We also added a search facility to this toolkit, which allows you to do a variety of different searches, like a substructure search based on the monomer DB, a subsequence or full sequence search on the macromolecule bit. You can extend your searches for specific residues, natural or non-natural one, or chemical modifications on your stored molecules. And you can also do a chemical substructure search on your registered large molecules. Let's have a look at the architecture of that toolkit. The center of all of it is a registration database. And this registration database kind of is closely linked to a so-called monomer database, which captures all of the individual building blocks, the templates you define. And these templates or building blocks you use to kind of create the larger entity biomolecules. The monomer database itself is a JChem base table, so we can do all of our chemical magic on the monomer database, chemical searching, substructure searches, full structure searches, fragment searches, all what you like. And we access this database by a set of core registration web services for registering new entities, registering or modifying registered entities and deleting entities if necessary. So this can be seen as the kind of core registration layer here. These core registration web services can interact with auxiliary web services. These can be converters, for example, between different formats. These can be calculators, as I mentioned before, for calculating molecular properties. The search service is an auxiliary web service. And you may think about certain connectors in order to integrate to other third-party applications where necessary. What we've done with this now is we created a kind of browser-based demo client, which directly interacts with the toolkit web service architecture. And this makes use of the Helm editor whenever it comes to the point of defining, editing, and modifying large molecules. And it makes use of our Marvin applet whenever it comes to the point of actually changing the content of the monomer database. As the Helm editor arises out of the open source project, the Pistoia Alliance, it was created as a desktop application, and hence what we have implemented additionally is a kind of update service which makes sure that the Helm editor always receives the latest content of the monomer database, which is centrally deployed. In the offline version, it would essentially only be able to use a monomer library, which is local on your computer. This is just a list of services. It may even be incomplete. I think it's nice if you want to go through the slides later on to have a read for yourself. What I'd like to highlight here is that we have a conversion facility which generates a mol file out of a Helm representation, or in fact any large molecule representation. And we are working on the reverse way as well, so having a mol file and then trying to interpret the biological sequence in case there is any in this molecule, and then adding the missing bits as chemical modifications, so to create a large molecule from a plain chemical structure graph. Where would you want to use such a toolkit? What I'm going through with you now are 3 use case scenarios, starting with the first one being registration or handling of peptide biotherapeutics, macrocyclics. The requirements for this are that I'm able to register sequence and chemical structure of these molecule class, that I'm able to achieve an unambiguous representation of the peptide sequence. Since it's cyclic, I have to find a canonical starting point for the cyclic sequence. And if I'm working in the field of developing these macrocyclics, I also appreciate the ability to quickly enumerate a library with residue replacements which are chosen from my set of building blocks at defined positions, which I have identified, for example, as the hotspot of activity of that peptide. This is a proof of concept which we implemented at a major pharma company with the help of our toolkit. And what I have to say right now is that I'm talking about the toolkit as a kind of integration toolkit. So it's really just offering you the web services in order to build your own applications. What I'm going to show you now, however, are some mock-ups, how the functionality I've just described to you, which was implemented in a custom-made application, could be used in one of our products, namely the Plexus Suite. For more information on our Plexus Suite, there is the webinar details happening on Thursday, 16th of October, as Janusz already mentioned. I'll just let it stand here for a while that the recording can capture it, and you can revisit it later on. So, as I mentioned, the enumeration of a random peptide library based on a starting sequence and having identified hotspots of activity. This is kind of a mock-up of our Plexus enumeration module, where we see here at the moment the sequence in question in the top left. We have a small panel where we can indicate which positions we actually will try to modify and mutate and enumerate. There are a few options about the enumeration type, and then you can set by the number of the residue the individual replacements you would like to see in your library. You have the option to kind of apply these for all or select individual replacements for the individual positions. These can come either from your monomer building block library. It can come from any chemical structure file, SD file, or you can draw yourself some new structures if you want. Once this is all set up, we can just hit enumerate. And what it does is it will provide you a library of peptides where the individual positions are a random combination or kind of permutation of all possible replacements you have chosen in the initial configuration. It can generate a few intrinsical molecular parameters, and you can then use this data set for further filtering in order to filter down those ones which are, I don't know, synthetically feasible or are within the boundaries of a certain range of one of its PhysChem parameters, for example. The second use case we are faced with is largely dealing with molecule registration. So people are asking us, can we deliver an agnostic entity or substance registration system? The requirement for this is that it needs to support domain-specific formats during the entity submission. So of course the biologist would like to submit a sequence or a FASTA file, whilst the chemist prefers to submit his entities in chemical structure format. That requires us to have a fast round-tripping functionality between these different representation formats, all under the aspect that this kind of interconversion between formats does make sense. So you probably would not want to convert a large antibody into a molecular graph because there's no real meaning behind that. But if you're talking about small peptides especially, then sometimes it is required to see them from the chemical structure point of view, and sometimes the sequence itself is of importance. Last but not least, that also means that it should be searchable from any query format, as I just mentioned. You may want to search it by the sequence, or you may search it by a chemical structure or substructure search. How our products operate currently is what I'm going to show you in this slide. So we do have a compound registration which integrates Marvin for drawing and then JChem base and JChem web services for all of the chemistry involved. And it accepts SD files, the common chemical file formats, and then stores this in a small molecule registry. There's then a clear boundary towards the biomolecule registration, as we do it at the moment, which is featured by the HELM editor, Marvin JChem base, and our biomolecule toolkit. And that accepts HELM or sequence or FASTA inputs, and it accepts mole or smiles whenever these are representing a biological molecule with standard residues, so standard peptide sequence with no modifications or standard nucleic acid with no modifications. This is then stored in a large molecule DB. What we actually would like to achieve is that we have a single input which goes into a perception engine, and this perception engine receives any file format and then tries to interpret the content of this file format, whether it should go either to the compound registration because it is said to be a small molecule, or to the biomolecule registration because it is an antibody, or even to both in case it is a small peptide, an example, where structure and sequence are similarly important. And that goes then into kind of databases or database tables, could reside even within the same database if it's required, making sure that they are stored in a way that the content is queryable meaningfully for both of the entity types. That's also offering you a way how these kind of large and small molecule databases are informationally linked. This is possible because the biomolecule toolkit in itself is offering a variety of file format supports, and also our kind of JChem and Marvin core is offering a similar support for different file formats, and the overlap between the two is the mole file. So as soon as we have kind of exported from our biomolecule toolkit a large molecule into a mole file, it can be taken over by the Marvin core and becomes available for our entire cheminformatic intelligence. The third use case, and with that the last one I would like to somehow mention here today, is antibody-drug conjugates, which are in the biologics world kind of a special case simply because you have a biological entity, an antibody or an antibody fragment, which is then coupled to a often chemical structure consisting of a linker and a payload molecule, which then as a total makes up your biopharmaceutically active substance. So the requirement for this is that you're able to register sequence, chemical structure, and eventually conjugated molecules, including post-translational modifications, that you're able to unambiguously represent the structure of such an ADC complex and also capture eventually relevant annotation reflecting additional structural annotations, for example an antibody-drug ratio, which tells you how many molecules of your conjugates do you have bound per molecule antibody. Since the antibody development part is to a large extent done independently of the payload development, you also kind of need an ability to quickly search on the kind of antibody domain or antibody chain sequence. And on the other hand, you need the ability to search on the linker chemistry or the payload you've added to your ADC. Also here, these are mock-ups, so they are not actual screenshots, but show you a similar approach one could take within the Plexus suite in order to run a design study generating new molecules by replacing, for example, a given linker which has turned out to be loaded with a suitable payload. So you see here that you need to separate the kind of antibody part in a different column. You have separate link outs of the linker and the conjugate themselves. The conjugation residue is known, the ADR may be known or unknown. If we are trying to kind of enumerate the linker here, we're going to click on replace. We can define the set of linkers coming from our building block library and generate the kind of new variants of this, which is essentially enumeration just with a variety of selected linkers from the previous screen. The value proposition of this toolkit, I think it offers value to two independent user groups. For the scientist itself, I think this toolkit can help you to share your molecules unambiguously internally or with external collaborators in your preferred file format as well, and making sure that everybody who receives this is actually able to understand it on the fly. So it saves valuable time on curating inconsistent representations received from partners or collaborators. It helps you to kind of generate libraries of biomolecules and do pre-filtering on these based on your own preferences, if you can make these calculations available from a web service, or we implement them for you. And as such, it can also help you to screen biological molecules for modifications and decorations, possibly helping you to generate better SAR models for the entities you're working with. For the people actually developing applications or working in the IT departments of organizations, one major benefit is that it may actually save you resources on maintaining parallel systems for specimen registration, since we aim to provide you with a system which is capable of dealing with both small and large molecules. It at the same time may remove your bottlenecks caused by legacy system limitations. I am aware of a number of instances where people are registering peptides into their compound registration systems, and a single registration, for example, may take up to several minutes because the tautomer check of the peptide chain is something which can take up quite some time if you don't have any workarounds for it. Whenever you move to larger proteins, of course, that scales with it. Whilst in our toolkit, we offer you instant registration within less than half second actually for any entity, including antibodies with a fully defined disulfide bridge pattern. And it allows you to create web-ready applications which are chemistry aware and biology aware for your R&D scientists. Regarding the roadmap, I've shown you the kind of 3 use cases which are in fact proof of concepts we are working on right now. And as soon as we've finished with these POCs, we intend to publicly release this Biomolecule toolkit as a preview version. That may happen still this month, but I think it's rather likely it will happen in October. Receive its product page, you'll find it there, the product descriptions by that time. In the mid and long-term plans, I think our focus will still largely stay on the integration bits and aspects, so that we need to implement a cell renderer, for example. If anybody wants to integrate our toolkit and needs to display these molecules in table-like outfits, then we need them to provide with a kind of nice graphical representation of these molecules. We have some improvements planned for better supporting ADCs and better supporting molecules with structure ambiguities. imagine in the case of ADCs that you do have a conjugate and you do know it's probably bound to a certain amino acid residue, but you don't know its exact location, or you have some mass spec result indicating that on a given fragment you have a modification which sits on one or the other amino acid, but you can't really tell which one it is, then it's something which needs to be found in the representation still, and we have to support the kind of additional data for this. I think in the long term, we also intend to kind of overhaul our Helm editor, which we currently integrate with an own sketcher, which will be probably JavaScript based, just as Marvin for JavaScript, which we offer for the chemical world at the moment. And there are a few other things going, and I think I should cut myself short not to stress it too much, because I would also like to show you some of this toolkit implementations in action. So I think that's the keyword. I should switch screen now. So maybe your screen will go blank for a moment while I stop sharing my screen and then reshare with the application reopened. There we go. You should now all be able to see my browser, in which I have loaded the demo client, as I've mentioned in my previous presentation. And what you see here is that it has, I left my configuration panel open. What you see here is that we have 3 tabs in the top. We are landing here on the Register Biomolecules tab, which will allow us to define new large molecules and then send them to the database. We have a monomer manager, which helps you defining the individual building blocks we can use in order to create our large molecules. And we have a search tab, which we can use to interrogate the molecules we have already stored in the database. What I would like to do first is I would just like to register something. Oxytocin, for example. It's a small peptide hormone which acts as a neuromodulator in the brain. It's a very small peptide, only 9 amino acids. So I'll copy this here into this input field, switch this to peptide sequence, and hit the load button. And you see each individual amino acid is now shown as a little box with a single amino acid letter code inside. And what oxytocin has a special structural features is that the 2 cysteines are actually bridged via a disulfide bridge, which you can choose here. And it has an additional amidation on the C-terminus. So I'm looking here for chemical modifiers, select the amine, drag it onto the canvas, and then attach it to the glycine. I can give it a name. And just select register. And this is very unfortunate. What's happening here? I'm not really sure what's happening here, but you know, this is still in development. I just add this to my notes, and let's go on with another example. Hopefully that works better. So what I have prepared as the next example is an antibody. Usually that example works well, actually. I will place the Helm notation of the antibody here, and you can see that it's actually quite lengthy. It's 2 heavy chains, 2 light chains, with the individual disulfide pattern. when I squeeze this into the editor, you may see that it actually looks relatively ugly, simply because this particular view of the HELM editor is not really well suited for working with antibody. But the most important message here is that we are sending this molecule now to our registration toolkit and store it in our DB. And you see it's almost instantaneously switching to done, which means we have it stored right now. We may get back to that later when I show you the search functionality. For those of you who are working with nucleic acids, I'd like to show you one example which will actually require us to register a few new monomers because they are representing special chemistry for this example I'm going to show. So I'm switching now to the Monomer Manager tab. And what I'm going to do is I'm going to register 2 new RNA monomers. I'm just pasting the name from a hidden cheat window I have open here at the same time. So if something's not happening, it means that I'm doing something somewhere else. I also paste the chemical structure, give this a abbreviation. The monomer type is RNA, and it's the functional type. This is a ribose, so it's a sugar. I have to specify some attachment points. This one's an H, R2 is an H, R3 is an OH. And then I can register this as a new monomer. I have a second one, which is methylcytosine. And we give this the abbreviation MC. And this is a nucleobase. It only needs a single attachment point defined. And I can register this. Now I'm going to switch back just to make sure that it's actually here. I can just type something here. And see, it's there. You see that it's actually registered for 2 monomer types. So nucleic acids are considered to be DNA and RNA, but the methylcytosine will be available for both types. So let's register the corresponding molecule. I will move this now to the editor. If I may. Otherwise I usually can. Oh, let's reload that page. Choose the HELM notation input. And you can see that the molecule now successfully imports. It would have missed the monomers if we hadn't done this. And you can see, if you look closely, that actually most of the individual monomers you can see are non-standard residue. So you have the entire backbone and stabilized with additional thiophosphates. You have these flanking regions of 5 nucleotides on each side, which carry the additional modification on the ribose unit. And then in the middle, you have some 10 residue long DNA sequence where you carry at 2 positions the additional methylcytosine. And this is an approved RNA drug, which is called mipromycin. I can register this as well. Last but not least, I have one thing which will illustrate the canonicalization options. So what I'm going to do is, I have here the structure representation in HELM notation of cyclosporin A. If I load this into the HELM editor, you see this is a circular peptide. And apart from 2 amino acids, it actually consists of modified amino acids, or even some very unusual amino acids. And can give that a name. And it should fail because I previously have already registered this. Remember that the ID, which is given here, is that it exists in our database under the entry CXN54. And what I'm going to do now is I'm going to grab, so I will delete the notation here. I'm going to grab the notation back from the editor, and I will change that notation by taking the first 2 amino acid residues and adding them to the end of the definition of this peptide. And I place this back into the editor. You see it has slightly changed, and you see that the A is no longer at position number 2, but it's now at 10. And if we register this, it will also tell us the canonicalization works. This molecule is already registered in the database. In the next part, I'd like to spend a few words on the search capabilities. So I'm going to show you here in an Excel spreadsheet what I've done. I've pre-registered a couple of peptides which we created using our enumeration engine. So the first thing is that we generated an alanine scan out of a kind of standard peptide sequence here, successively replacing each individual amino acids with an alanine. In the second series, we have replaced each individual standard amino acid with its D variant, its stereoisomer. And last but not least, the last 3 molecules are having a disulfide bridge between different cysteines of that sequence. That's just important to know to understand what we're going to see here on the search page now. So on the search page, I do have a query builder, which kind of on the API level works modular, but we've only implemented the kind of single level query capabilities here at the moment. So you can define a sequence. I choose MTG. You can define a match type, full sequence or subsequence. I can even limit it down to I'm interested to only peptide or RNA or DNA. So I'm interested in peptide. And I hit search. It shows me a relatively long list of peptides. But what I can do now is I can choose a modification which affects the kind of subsequence query I've defined here. And say, please show me everything which has unnatural residues included. So it limits down the number of hits we get back, and you see that only those ones where actually an unnatural residue is within the molecule are retrieved back as search results. If I look for molecules which carry a chemical modification, then it will look for all those molecules which on their kind of backbone have additional modifications which are outside of the regular N to C bonding pattern of the backbone of the peptide, or nucleic acid if you want. So in this case it captures all of the disulfide bridges, and it captures also the molecule which has a terminal acetylation. On the API level, we can also tune this to look for specific chemical monomers. If I am looking for the D variant of an amino acid, I can specify this, and it finds back only those which are carrying this exact monomer. And we are also looking to implement soon kind of numerical options here, that I can look for sequences which carry a certain number of chemical modifications, even if you specify them. For example, I'm interested in the number of phosphorylations, phosphotyrosines, for example, in a protein. But let's go back to the unmodified example. I take this molecule, and what I would like to demonstrate now is kind of the way how you can round trip between our biomolecule toolkit and our chemical platform. So I'm selecting this peptide, and I hit this wonderful export to mol button here, and it will export this molecule into a mol file. Somewhere I should have a Marvin sketch window open. I can bring this in here. And all I do is I press Control V so that the structure shows up. Now, the specialty is that when I hover over the chemical structure, you will see that the individual residues of the sequence are preserved as superatom groups. And from here on, I can now do all of my play all of my cheminformatics capabilities if I wanted to. But let's see if we can do the reverse as well. So what I have prepared for this, clean this. I will open the same sequence. And you see there are no superatom groups defined. In order to make sure that you actually do believe me, what I'm going to do is I will see this as a MDR mol file. And I will re-import this. As we all know, the mol file does not carry along sequence information. So the layout has changed a little bit, but it's essentially the same thing. And now our kind of core chemistry module offers you the option to convert this, if it's a standard sequence of, if it's a sequence of standard amino acid residues, to convert this back into a peptide sequence. You can see this here in this window now. It recognizes from the chemical structure the peptide sequence. And this peptide sequence I could now feed into my biomolecule toolkit, which can convert it into any other biological file format, and then use this for registering this molecule from a chemical structure format. Last but not least, I'd like to go once more back to the search page here. Oh, that was a bit too much. I should use a slightly longer sequence. You remember the cyclosporin A. You see that it has a number of kind of unusual amino acids. What I'd like to show you now shall somehow illustrate that, first of all, we've linked our monomer library to the large molecule library. And second of all, that we use the same functionality to create the regenerated Helm notations for enumerating our peptide libraries, for example. So I went to the monomer manager. I look for the methylated BMT because I know that's one of the monomers in the cyclosporine A. And I'm going to change this abbreviation to something else. And now I hit update this selected monomer. It tells me it's done. Now this can be a potentially dangerous step because you use this monomer, which is kind of part of a defined macromolecule. So let's see what happened on the data set here. Again, I'm looking for the base sequence of the cyclosporine A. And you see here that it actually has regenerated from updating the monomer also the notation of this large molecule, which means that there's a replacement happening on the kind of peptide sequence, if you want, and we use this function for the library enumeration. And at the same time, we can use this for making sure that whenever you change something on your building block database, that it actually reflects or is reflected in the database of your large molecules. We do have a configuration option which would prevent you from modifying any monomers which are already used to define a large molecule. So that's purely a configuration switch you have to set in the deployment process. And I think we are approaching the hour. This is all I'm going to show you today. I very much appreciate your attention. I hope you learned something. I hope you had some fun. And I hope you'll bombard us with questions. I'll give it over to Janos.